Bacteriophages, or simply phages, are viruses that infect bacteria. There are two basic types of infections that bacteriophages can cause on their bacterial host. Some bacteriophages are lytic, meaning they will cause lytic infections. Others are temperate phages, meaning they will cause what we call lysogenic infections. We're first going to look at the lytic viruses, which cause lytic infections. Lytic viruses go through lytic replication. This is a productive form of viral replication because viral particles will be produced as a result of this lytic replication cycle. An example of a bacteriophage that goes through the lytic replication cycle is T4. The host for the T4 phage is E. coli. Burst time is the time that it takes for the virus to infect its host, E. coli, go through lytic replication, and produce viral particles. In the case of the T4 phage, the burst time is 30 minutes. Lytic replication has 5 steps: attachment of the phage onto the host, entry of the virus's genome onto the host, synthesis of viral proteins and replication of viral nucleic acids, assembly of the viral proteins produced during synthesis, and finally, release of the newly assembled viral particles. The first step, attachment, occurs when the phage contacts the host through random collisions or Brownian movement. Viral particles are constantly being bombarded by air molecules, and through these random shaking, the phage may encounter the host. In the case of the T4 phage, its host is E. coli. Attachment of the virus to the host is dependent on the precise fit between the recognition sites found in the filament fibers of the phage and the specific attachment proteins on the surface of the host cell. Remember, these attachment between viral recognition sites of the virus and specific receptors in the host is what determines viral specificity. Here is the bacteriophage bound to the host receptors. The second step is genome entry. In the case of bacteriophages, the nucleic acid of the virus is injected into the host. The empty capsid of the virus is left outside the host, and it will eventually be disintegrated. The nucleic acid of the virus is injected into the host as the phage tail sheath contracts as a syringe, forcing the nucleic acid of the virus through the cell wall and the cell membrane of the cellular host. The next step is synthesis of proteins and replication of the viral genome. Remember, the virus does not have any organelles. Viral protein synthesis will take place thanks to the cellular ribosomes. First proteins that are going to be transcribed and translated will be the viral proteins that helps the virus take over the host. These early proteins will be transcribed and translated within minutes after infection. Proteins like nucleases, which will degrade the host's DNA into individual nucleotides. These will accomplish two things. One, it will provide nucleotides for replication of viral DNA, and it will prevent the cell from transcribing and translating its own proteins. other early viral proteins are going to modify the host's RNA polymerase so as to recognize viral DNA. Late proteins are proteins that are transcribed and translated later during the infection. These are going to be structural proteins that make up the capsid and the tail of the virus. Protein synthesis in this case is similar to cellular transcription and translation, except that the messenger RNA that is being transcribed is a copy of the viral DNA, not the cellular DNA. Therefore, the proteins being translated are viral proteins. The replication of the viral genome is also taking place, so multiple copies of viral DNA is being synthesized. Remember, at this point, the cell genome has been destroyed by early viral enzymes. These viral particles that have been produced are going to be assembled into a multitude of viruses. Next to the last stage is assembly. That happens when the viral particles that were produced during late transcription and translation are assembled. Each of the little viral particles will be assembled and it will receive a copy of the viral DNA. The last step in the infection is release of the virions from the host cell. These involved late transcription of lysozymes, which are produced from the viral DNA. These lysozymes will digest the cell wall and the cell membrane of the host cell, releasing the viral particles and essentially killing the host cell. Burst size of the T4 phage is about 200 phages per infection. The burst size is the number of viral particles being produced through the lytic infection. Remember, burst time is the time that the lytic infection takes from beginning to end. Burst size is the number of phage particles produced during the infection.